Hey everybody, in this video we are going to talk more about introns. I would like to start though with some questions here that might help you think about exons and introns in a different way. So, or let's say a couple questions. So, are all mRNA coding sequences found in exons? So remember to watch the previous video to learn what is an exon and what is an intron. What do I mean by exons and introns? Check out the other video if you are unfamiliar with those terms. So are all mRNA coding sequences found in exons? And for that, the answer would be yes. Yes, okay, so all coding sequences, we expect them to be found in exons. So they shouldn't be found in introns. Introns are removed. So how about the opposite question? Let's see, are all exons made up of coding sequences? So are all exons made up of coding sequences? Do all exons contain coding sequences? And for that, if you were paying attention last video, it would be no, right? Because there are 5' untranslated sequences or untranslated regions of messenger RNAs and 3' untranslated regions of messenger RNAs. So, and these are important, even though they're not translated. They're important for regulation of gene expression, such as, say, transport of that messenger RNA out of the nucleus and into the cytoplasm. Also ribosome binding, stability of the messenger RNA, how quickly it is degraded by, say, nucleases in the cytoplasm. All that stuff, is influenced by the 5' UTR and 3' UTR. So we can see how mutations in the 5' UTR and 3' UTR, or in the coding sequence, mutations in the coding sequence could change the amino acid sequence of the protein. So mutations in the exons could alter the function of that messenger RNA or that protein after translation of the messenger RNA. So what about intron sequences? Are intron sequences important? So, we have to say some parts, yes, right? Some parts of the introns are important. And some obvious parts are the 5' consensus sequence, next to the 5' splice site, the GU, or GT if you're thinking about the DNA sequence corresponding to the intron, or the 3' splice site, the 3' consensus sequences found next to the 3' splice site, which is AG, right? So those are pretty key. We don't want those to be mutated, otherwise splicing of the exons would not work. Splicing of the exons flanking that intron would not work. And there are other parts that are important for splicing, and I'm going to show you that even if we don't understand exactly how splicing works, there are key residues within introns that can influence the splicing pattern, and that can have a major effect on the resulting messenger RNA and the protein. And then there are other parts we're just not quite sure about yet. And I alluded to this in the previous video. Why are some introns in humans and other organisms so long? Why are they so long? And we don't have a good answer for that question yet. But in this video, there is an interesting paper where there's a mutation in an intron and it leads to a genetic disorder. Now, that paper is I guess if you wanted to look it up. And it should be freely available to ISU students. So Guaquilin et al. 2018 in their journal Brain, volume 141, number 1. And there's this little E1. I guess it's possibly this might be a internet-only journal where they don't have an actually printed issue, but this might be the first volume or the first article in volume 141. The first article in volume 141, issue 1. So article E1. Anyways, what is the title? So the title is POLR3A variants in hereditary spastic paraplegia and ataxia. So, okay, HSP, this is a genetic disorder. So there are like 80 different types of HSP, hereditary spastic paraplegia, 80 different types, and the major I guess sign or symptom of the disorder is difficulty walking, and there's some neuron degeneration, and it's particularly the neurons that help individuals walk. So in this article, you know, they claim they have found that mutations in a RNA polymerase III subunit. So RNA polymerase III. Now RNA polymerase III, that's involved in transcription of transfer RNAs in one of the ribosomal RNAs. And it's got many different subunits. One of the subunits is POLR3A, encoded by the POLR3A gene. And, you know, they make a pretty convincing argument that there is, if you can get a mutation in here, if you have a mutation in here, well, then you can suffer from this disorder. So, this is a relatively recent paper, as you can see here, 2018. And I like it because, you know, I think it's particularly relevant to our discussion of splicing of exons. And it's a human example, which I like to talk about. So let's just, and they have a nice pedigree in this article as well. So let me walk you through the pedigree. So we have two parents here, mom and dad. They both have one wild type allele. So the genotypes I'll be showing you are the genotypes for the POLR3A gene. Now they have a wild type allele, and the dad here has an allele called C.91C to T. Now what does this mean? Essentially it means codon 91 has a C to T mutation. And the mom over here has an allele that's called C1909 plus 22G to A. Now, what does this mean? Okay, so codon 1909 is a pretty big gene, or messenger RNA, that is. Codon 1909, go to that one, then count 22 bases in, there's a G, change that to an A. So that's this allele encoded by mom. And essentially, you know, I don't really like the way that the alleles are described, but with this one, they need to do it this way because this is in an intron. This mutation is in an intron. And I will show you more specifically the sequence of this intron in a second. So these two individuals had children, 7 kids to be exact, and so I think this should be F2, 3, right? Let me see. Let me check the notes over here. So these notes are, again, are available on ReggieNet. No, I don't know why they call this, in the article, I think they call this F1-3. I would have called this F2-1, being the second generation. So this individual here has the disorder HSP. This daughter does not. This son has the disorder. This son does not. Daughter has the disorder. Son has the disorder. And this son apparently passed away, and we don't have any genotype information for that individual here. So that's 7 individuals here. This one's F1-5, F1-6, F1-7, and F1-8. And let's look at the genotypes. C91, C to T, and C1909, plus, this is a pain to write. So here, this individual has 2 mutant alleles. Inherited this mutant allele from dad, this mutant allele from mom. We're going to call these compound heterozygotes. Can't call them homozygotes, right? Because the mutant alleles are different. They have 2. This lady here has 2 different mutant alleles. They're not homozygous. They're not the same alleles. So we're going to call her genotype a compound heterozygous genotype for containing 2 mutant alleles. This one is wild type with the C91C to T mutation. This one, do you think this man here is going to have or male is going to have a wild type allele or 2 mutant alleles? 2 mutants, right? It has the disorder. So this one and this one, I won't diagram, I'll just point. This one and this one. This one right here, 2 wild type alleles. This one right here, what do you think? 2 mutant alleles? Yep, 2 mutant alleles. One of here, inherited this one and this one. How about this one right here? Same thing, 2 mutant alleles. Inherited this one from dad, this one from mom. And this one we don't know. Okay, so you know, we have a pretty convincing correlation between having this disorder and having 2 mutant alleles for this gene right here, POLR3A. So this one right here, this is in an intron. This mutation up here, the C to T mutation turns a coding codon into a stop codon. So it ends up making creating a messenger RNA that codes for a very short and non-functional protein. This one right here, though, the mutation in the intron, so why does the mutation in the intron influence function of this gene right here? It must not be completely functional, right? Because we can see this individual here who has a wild type allele in the C91 allele. Well, this individual is perfectly fine. She doesn't have the disorder. So whatever is wrong with this allele, with the 22G-A mutation, it must be less functional than wild type. So I have in the notes, I kind of, you can download the notes and you can see what this mutation does. So we can cross that out. Okay, here's the C1909 plus 22G-A mutation. So this is the normal allele, the normal sequence. And what I'm showing you is the intron. It starts with the GT. We're looking at the DNA sequence instead of the RNA sequence. We're just looking at looking at the coding strand of the DNA sequence. So GT, intron start, AG, intron stop. Or Yes. So the intron's much longer than this, and I have these little 3 ... dots to let you know that I'm not showing all the sequence of the introns. In this one, if this was in color, we could see that this is a G. So the normal sequence has a G at this position, and we can count. So let's say this is codon 1909 right here. So let's count 22 in. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20, 21, 22. Okay, change that to an A, and you'll have our mutant allele right here. So all the sequence right here is identical to the sequence up here. And what I'm showing you is that this mutation here, what the authors of the paper found, was that changing this G to an A moves the 5' splice site all the way down here. So it moves it all from here all the way down here. And as a result, when this is this becomes part of the exon. So the exon length, this exon here, its length increases in the mutant version of the messenger RNA. As a result, when it is translated, we get a protein with a different sequence. And I believe there is a frame shift. This causes a frame shift mutation. So we end up with a longer exon and the reading frame changes, which is something we're not going to talk about until next week. So I don't want to talk too much about that. Frame shift, I don't want to talk too much about that in this video because we talk about frame shift mutations when you return from spring break. So this is interesting here, how we have a mutation here and somehow it changes the location of the 5' splice site and increases the length of one of the exons. So this is just an example of how a mutation in an intron, even though it's not a coding sequence, can influence the function of the gene. So it helps emphasize the point that, at least some sequences within introns are important or critical even for the function of a gene. So I hope you liked that example of introns and humans and how the sequences are important for normal function of the gene and how they can lead to a genetic disorder. So here's an interesting question too, if you're curious. So RNA pol 3 is in charge of transcribing tRNAs and a ribosomal RNA, one of the ribosomal RNAs. And it's an important enzyme. We can't live without it. So the authors think that mutant allele with the funny exon, it thinks sometimes the messenger RNA is spliced correctly and you get functional protein. Otherwise, an individual with 2 non-functional pol R3A alleles is probably not going to be viable. But so the authors think that having that less functional allele, C1909 plus 22G to A, that is sometimes spliced incorrectly, they think it causes problems because RNA pol 3 is in charge of tRNA transcription, and it is known that when we have problems with transcription of problems with tRNA levels, so the nervous system is one of the first systems to be affected. So one of the first systems to suffer from low tRNA levels or abnormal tRNA levels. So we have a connection between mutation in pol R3A, which alters the function of RNA pol 3, which is important for tRNA production, which could influence the nervous system and lead to symptoms of HSP, which the primary symptom is difficulty walking. Okay, so I hope you like that example, and I'll see you in the next video where we talk about alternate splicing.